has a bachelor degree in genome sciences or genomics from the Universidad Nacional Autónoma de México, UNAM, for those that are from UNAM, and a PhD degree from the University of Cambridge in the UK in cancer genetics. After the PhD, Daniela was a postdoctoral fellow at the Experimental Cancer Genetics Group in Wellcome Trust Center Institute. Currently, Daniela is an assistant professor at UNAM, where she is a group leader at the International Laboratory for Human Genome Research, focusing on investigation associated, sorry.
Tenure last week.
Tenure last week. Oh, very good. Very good. Okay, let's go back. So she's still a group leader at the International Laboratory for Human Genome Research, focusing on the investigation of the oranges, mutational drives, and potential therapeutic targets of cancer types that are important to Mexico and other Latin America countries. So she's important to all of us. In her lab, they use analysis of next-generation sequence data, both DNA and RNA, from tumor and germline matched samples from Latin America patients. That was something that I could not change from her presentation, from her introduction in her website, because it's impossible. I don't know what means germline matched samples. I hope it's good. And now I'm going to introduce Mariana Gomez-Schiavon. Professor Mariana Gomez-Schiavon has a bachelor degree in genomics, genome science, also from the same place as Daniela. I think you studied together, right? That's amazing. Long time ago. Not so long, come on. And Mariana has a master's degree in biomedical engineering and physics. Okay, yeah. Centro de Investigación en Estudios Avanzados del Instituto Politécnico Nacional, right? Oh, that's good. Unidad de Monterrey, for those who know where it is. And a PhD degree in computational biology from the Duke University in the US. In sequence, Mariana was a postdoctoral researcher at the University of California, San Francisco. Not bad. And currently, you are, what your status now? Assistant professor at UNAM in the same lab of Daniela. In her research, Mariana's interest in understanding the dynamic properties of gene regulatory circuits, how they emerge, how they proliferate, and persist through natural selection. Her work combines evolutionary theory, population genetics, biophysical models of gene regulatory circuits. So it's an amazing job. I'm very excited about that. So help me to welcome these ladies.
Hi, yes. Oh, thank you, Flavia, and thanks everyone here for the invitation to this course. I'm very excited about coming here and talking to all of you. I read all your biographies. It's amazing what you guys are doing and are interested in. And we're hoping that this course helps you think a little bit about what we do when we analyze data. I think many of you may, in your future paths, come across data. It could be in physics, it could be in biology, or in other areas. And we're hoping that this course is the first time we give it, because it was made for this class. So you can also tell us what you want to discuss, if you want to go deeper into any of these topics. But we're hoping that it goes into the fundamentals or the generalities of just any data research driven project, and what we have to take into account, where pitfalls may arise, what can we do about statistics, for example, that is really important in data-driven research. And we're also hoping to have a very active learning experience for all of you. So loads of activities, loads of questions, team activities, papers, discussion. So yeah, we're hoping you enjoy it. And I'll leave you to start with Mariana. Does it work?
Yeah, so I hope it's working. Yeah, it's working. Okay. So in the same direction, please interrupt in any moment. Questions are more than welcome. I think you're going to see this in the next 2 weeks. Both Daniela and I tend to talk a lot. So please, please interrupt us at any moment. If it is not clear, if you want to add something, I think if this is more interactive, we all enjoy it more. So that's the idea. Yeah, we... Yes, please. So you can see the idea of the course. Perfect. Okay, so that's perfect. Should I move it? Let's see. Okay, so we just wanted to do a really quick go over what was the idea for the class. I mean, Daniela already did an amazing job telling you the general idea, but here just in a little more detail. Our objective for this class is more than anything that you understand what is, in the very general, big picture kind of style, what is data-driven research, and then that, of course, requires you to learn the basic concepts associated to it. We are going to focus a lot of time in discussing the principles of data generations and statistical analysis required for this data-driven research, specifically in genomic science, because that is our background. But I think many of the things you're going to learn here applies for any big data kind of thing. And we are also going to spend a lot of time reflecting about important pitfalls and complications of data-driven research. And we decided to do this way because I think data-driven research is a very kind of open-ended kind of thing that you can do, but then there are also very clear things that you can do wrong. So I think if you learn what you should not do, then you can do whatever you want, right? So we are going to focus on that part. And just to be clear here about the philosophy and what we think this class should be, we believe, and I think there is another person from our program here, from undergrad program here, so that's how they taught us. So we are also going on the same philosophy. We believe that each of you is here because you want to learn more about data-driven research. So we expect you to do the best to take advantage of this course. And of course, we are also committed to do our best. So I think that comes with communication. If something is not working, please let us know, and we do our best to fix it. And like the more essential expectations for you as participants is, of course, we expect you to be here. If anything happens, we expect you to let us know why you're not going to be here. This is also important because we have a lot of team activities. So it's important for us to know if someone is not coming. I think they say that you're checking cards like all the time, like they are very strict with punctuality, and we are going to do the same. We are going to try to take advantage of the whole time. We have a very packed schedule with all the activities. So classes will start on time. And I think this is also to be respectful of everybody's time. Participation is very important. Please be present and active during the class. I think this is essential to really take advantage of what we have planned. And of course, respect. So everybody needs to be respectful to everybody else. That's a very, very important rule. We need to listen whenever anyone else is talking. And of course, criticism is always welcome if it is constructive, right? Okay, good. So that's it. Just to give you an idea of what we had planned for the class, all the yellow things are activities, and only the blue ones are like more lecture style, also with discussion. But we have a lot of activities planned for you. We are going to share this with you, so don't worry too much about the details right now, just so you have an idea of the different topics. Today is going to be just an introduction. Tomorrow we will focus on data generation, the details about what you need to think about when you are generating the data or using data from someone else. The third class is about the statistics. What do you do with that data? We think also that a very important part of data-driven research is actually find the right way to visualize the data. So we are spending the whole 4th day on that. We then are going to focus more, as I was saying, the pitfalls, the problems here. So we are going to talk about the narrative potential and how that can actually trick us. We are also going to spend, I think this is more general than just data-driven research, but very, very important. There is like some pressure from scientific community that can bias your results, and being aware of that bias is always the first step to kind of counteract it in some way. We are also going to spend our whole class talking about reproducibility crisis. That's a very important problem in science in general, but particularly in data-driven research. And just because we didn't want to be all negative, Daniela is going to finish telling you a little bit of some success stories on data-driven research, just to say like, oh, this can also be very powerful and useful. The ninth day, we are just going over the whole thing as conclusions to close over. And then the 10th day, you're going to have the final activity. So the final activity is also by groups. You are going to be discussing and presenting a different paper. So this is actually something I wanted to show you in more detail. Let me see. Here it is. Okay. So I'm going to put the link in a moment here so you can see it. But basically, in that page, you are going to put, actually, I can click it. Let me see. Ugh, I don't know what it said. Oh, sorry. OK, it was working. It's just not my computer. OK, so you're going to access this. You're going to put your name. And you're going to choose your rank, your favorite fields. And from that, I'm going to try to make teams to give you priority to whatever you prefer. And then basically, here it is. From that teams, I will share with you all the papers you're going to discuss. Actually, we have 2 to 3 options for each of the topics. So you can choose one of those papers. And you don't have to really read it ahead, but if you want to, it's going to make things for you easier. But the idea is that last Friday, the last class, the last session, teams will have 30 minutes to join and discuss the paper. Of course, that usually is not enough to read the whole paper, but that's OK. It's not the idea that you're going to explain the whole paper, but focus on what you learned on the class and how it applies to the story you are seeing there, discussing specifically what are the statistical approaches they are using. Again, not the technical details, but the big picture. What are their reproducibility practices, etc. We chose the papers because some of these are not our fields. We are not saying these are the best papers. We are not saying they are doing things right. It's part of the idea of the teams to actually discuss it. And you're only going to have a few minutes. I put it, oh yeah, 5 minutes, it says there. You're going to have 5 minutes to present it. So again, 5 minutes is not enough to present the whole paper. The idea is to make the important points clear for the class. And we are going to be very strict about the time, so we have some questions at the end and some discussion at the end. But that will be your final activity. And yeah, here are the names of the papers. We are going to share this link with you. And I think that's all.
Awesome. Ken, yes. Thanks for that. So, OK, so that's pretty much the setup of the course. Any questions about that? No? Happy to start. OK. So let's see. View, presenter, view, I guess. Is this? Yeah. OK. So today is introduction. How much time do I have? Is it 20 minutes? It's just we have a very tight schedule here. And is this working? Let me just see. Sorry, guys. I want to walk around and just to see if this is working. Is it? No. Wait. Ah. No? Sorry, wait. Where is this? Well, if not, I can comment. I wanted to advance this. I think it changed. It changed before, but I'm not sure it works anymore. If not, it doesn't matter. I can come and click it. OK, so today is the introduction. And today we're going to think about really thank you so much. What really is data-driven research? I think it's a bit different maybe from what you have been learning on this course so far. And today we're going to think about how it's different and how it really affects our conclusions from our studies that we conduct in this way. This is a different one. It's mine.
I think that OK.
So let me just see. Yeah, I think you fixed it somehow. OK. No, thank you. OK, so I've written here 3 different definitions of what data-driven research is. So basically, exploratory approaches to analyze big data. Big data that's being generated at massive speeds, amounts staggering amounts in biology at least, but in many other areas as well, for example, astronomy and social media, I don't know, all these other research styles that require analyzing loads of data. Now, what is the objective of it? We want to extract scientifically interesting insights, of course. So we have lots of data, and what is the data telling us, right? And the way we do those things is basically by discovering correlations, for example, querying it, trying to identify which of those correlations are useful for what we want to determine, and also finding which of those correlations are actually important, right? Because you will find loads of correlations in your data, and how did you determine which ones are important is really the goal of data-driven research. Okay, so any questions here? Is that clear? Right? Okay, so I want here for you guys to tell me, you've already taken a course called hypothesis-driven research. I saw. So please tell me, what do you think, based on what I told you just now, what is the difference between data-driven and hypothesis-driven research? Who has ideas? Over there. Oh, nice, you have. It's good that you have a mic too. I didn't know that. What's your name so I can start knowing you?
Thales.
Tanis?
Thales.
Tanis, okay.
Well, I suppose that I don't think it's like a rule, but I think usually in data-driven research, maybe first you have the data and then you think about questions that you can ask with that data. And hypothesis-driven, ideally, you first came up with an idea of what you want to test, and then you do the data collection accordingly.
So it's a bit like a reverse situation. Yeah, I agree with that. I think there's many ways of viewing that. I think that's, accurate. Any other ideas? Is there, or everyone agrees? Yeah? So, okay. Basically, this is probably what you because I wasn't in this course, but this is probably what you saw in data-driven research, right? And this is what, can you tell me again your name? Tanis, yes, I will, sorry, I will remember all your names, Susie. Okay, so first, yes, indeed, we derive research questions from what we already know from the world, or what we have observed, and then we come up with a hypothesis, and then design studies to collect data and try to confirm or refute that hypothesis, right? And this is also known as very classically the scientific method. This is, classical research, how it's been going on for many, many, many years. However, here, the data-driven approach, basically, yes, he was right. You first obtain sources. You do have to have an idea, of course. It's not just any data, right? You are interested in gene expression in certain disease and what genes are important for this, a healthy state versus a disease state. So you are interested in something, but it's not defined yet. You know the direction of your idea, right? So you identify what you want to know in general, and then you obtain sources of data. And then this is the critical step, and where we spend most of our time. You have a question? Yeah. And can you all say your name first at the beginning so I can start learning them, please?
Hi.
Yes.
My name is Andriele, and I think it's a basic question. What's the difference between data-driven research and meta-analysis? Is the same thing?
So we will also touch on meta-analysis in this course later, but in meta-analysis, this has to do a bit with the reproducibility bit that we're going to talk about. But in meta-analysis, you get together several sources of not exactly data, but usually studies that have been done on data, but you have already their statistics, for example, their summary, their conclusions, and you try to combine them to see if the direction of whatever you're looking at, for example, is this gene, well, maybe this process more associated with certain disease, because you have many studies, and some of them tell you, it's very associated, and some of them tell you maybe not that much. You combine all of that and trying to figure out what the truth is. Right? So it is very related to data-driven research, but you combine what other people have already done usually, right? These many studies with a lot of data, some of them very big, and you try to increase your your N, your number of subjects that are studied. But that is also tricky because people have analyzed data in many different ways, and you have to be very careful, you know, when you combine them. But basically, it's that. It's putting a lot of data together, even more than one these individual studies, and try to find out the truth. And we will touch a little bit on that, a lot more to the reproducibility stuff, but we will discuss that later. Is that okay? Any other questions for that? Do we continue? Okay, so basically, this bit, cleanse, extract, annotate data, it takes a very, very long time, guys. I mean, I'm sure many of you have already worked with data, and it's the most important step. If you, as they say in English, garbage in, garbage out. If you don't have your data clean, if you don't know exactly how your data looks like, the patterns, how it's distributed in many variables, you will just confuse yourself. And you will take even more time when you were like, oh, actually, you know, I should have, you know, removed this variable that was not specified in all the, I don't know, subjects. No, so this bit takes a very, 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 very long time, like 80% of the time, just saying. It's my trauma speaking. You know, I did a PhD for 3 years. I just cleaned data. But it's normal, it's normal. I thought, you know, when you're going through this, it's like, oh my God, it's me, like it takes me. No, this is normal, I'm saying it, you know, generally. So if you work and you take a very, 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 very long time, yeah, that's expected here. And then you analyze it. And we are gonna talk about how you analyze data. Usually with big data, you need statistics, very strong statistics, and statistics is very complicated. Like, you know, there's many, many different kinds of p-values. Who has heard of p-values here? We're gonna have a whole class on p-values, because what do they mean? Okay, that's class 3. We're gonna talk about that, but that's important. And then finally, you make a model, and then that's the end of this step. Of course, that's not the end of your research. Like you find a pattern, you determine that pattern is important, and that's, you know, just the first step to your actual next, you know, confirmatory research. Okay. And, you know, I just put here some pictures of, you know, many areas that are using massive amounts of data. No, genomics is one. Genomics is generating like whole genomes and transcriptomes and proteomic sets at a speed that it has never been seen before. And there's a lack of people to analyze, you know, these data sets. Also, in astronomy, as I was saying, and all these physics projects that, you know, are trying to investigate fundamental physics particles, for example, all the amount of information they generate, and they require, of course, these very strong statistics. Also, social media. Lots of companies now use all these, you know, every click and everything, and then the Twitter and the Facebook and everything, to do things like targeted advertising and things like that, right? And that also requires loads of statistics. And then, well, finally, of course, government surveillance. It's a real thing that happens. And of course, all the data from cell phones and localization, right? And, you know, I honestly think there's a tendency now that loads of areas of research are just turning to analyzing data. So I think it's important to know these distinctions. Any other ideas, actually? Do you know of any other projects that use, or not projects, but like areas that use massive amounts of data? This is all I could think of. I actually don't know. There must be many. Well, I think you get the idea. Okay. Right, so to do data-driven research also, you require a lot of expertises, right? It usually is an undertaking that's done by a team of people. It's hard, maybe impossible, for one person to dominate all the areas that you need to know in order to undertake a project like this. So first, we need database management, because obviously all this data is stored in databases. And the way you retrieve data efficiently, if you want to see, you know, patterns and all this data, you have to retrieve it fast in order to, you know, do whatever question you're looking for. So you need people that know, you know, efficient querying, indexing, retrieval, storage, basically IT systems. So that's one of the most important areas that's crucial when we undertake data-driven research projects. Also, we need data mining. So now we have the data, how do we analyze it, right? We need statistics, we need classification, we need visualization, we need detecting patterns, the actual analysis techniques. And then finally, well, you have loads of correlations, right? How do you find which ones are important for the question that you are exploring, right? So for that, and we'll talk about that, patterns, correlations, and how do we determine what's important. Any questions here, more or less? Let's get in there. Let me just see what the time is. Okay, yeah, we have time. Yeah? All good? Okay, now. Here it's very interactive. I want you to tell me what you think. Here, I want to put some questions that this person, Sabina Leonelli, she's a professor of history of science in Exeter in the UK, and she has done a lot of work. She's recognized for thinking about these questions, right? What does data-driven research mean in the world today? And I got some questions that she's, open questions that I think we should think about before like going and delving into, what the fundamentals of this is. So this is for you to tell me first. What do you think about this? Does our reliance on big data change the very idea of biological discovery and what counts as biological knowledge? What do you think? So how did people do research before and how do they do it now? And does that change what a biological discovery is? Again, yeah.
Well, I don't really know the answer, but I think that's maybe more a matter of scale, because before, because when you say that like data-driven, you have to deal with a lot of data, and then you can ask, okay, how much is a lot? And you cannot make a clear cut. So I think that before computers and everything, there were some people that dealt with a lot of data, but they of course used to do that according to the limits of what a human can do. So like great revisions and things like that. So I don't think it changes the thing in the essence, but it's a different way of doing things, because then you have to know a lot of skills like computation and other things that are not used or not used in this past great research that used to do a large revisions with many kinds of data.
Mm-hmm. Mm-hmm. And what's your name, please?
Hi, my name is Kauye. So what I thought regarding this question is that biological discovery was frequently, and still today, it happened frequently as, for example, an image which shows a cellular process, or very episodical kind of register of evidence. And in a sense, I think it can biological discovery with data-driven may mean a stir towards a more statistically relevant kind of discovery, because sometimes cellular imaging may be registering an anomalous thing and not really a new process, and it is frequent. I mean, I'm thinking about molecular biology, but I think that generalizes to many other areas inside biology too.
I agree. I think some people, people think about these things, right? And there's criticism. And it goes related actually with what you two said, because some people said, well, in the past, people thought, you see things, make some experiments, make a hypothesis, really think about, what is happening, I'm going to test it. Oh, I find something new, right? Like, this is how it works. And now, and I say this as a person that does data-driven research, this is my thing. You have all this data, and exactly what you're seeing is some correlations, and maybe you find something, but is that big? Is that meaningful for biology? Is that really a biological discovery, or is that? Is it? I do data-driven research, so I think some of them are, but some of them, I do agree. Maybe you see some papers, and it's a bunch of data, and then, but what? what really did I learn from this? I mean, I think that's at least what some people think about these questions, right? Like, is it changing really? Also related to these answers and these images, right? Before, biological discoveries were done maybe by a small group of people. I mean, I don't think this like the lone genius is actually true, but it was a small research group, right? And you get the data and you think about it. And now, I mean, how many authors do papers have? And people do all these things. I mean, they are important, like data collection. And in the case of like hospitals, like all the people that have to collect, you know, consent the patient, process the sample, getting, you know, data storage. So it also has changed really how, you know, biological research is undertaken, right? So I think this is provocative, right? What counts as biological discovery? And we can also discuss that in one of the next questions. Anyone wants to say something else about this question? You agree? Very good. I have another one for you. Okay. Actually, Leonelli has another one for you, not me. But I think it's interesting. So what role do theories play in data-intensive research? And how does big data biology relate to hypothesis-driven, observational, and exploratory research? So have you seen what observational and exploratory research are? Have you seen it in one of your courses? Not yet? Oh, sorry, so fine. Well, hypothesis-driven you've seen, you know that. Observational research usually refers to seeing things in their natural environment. You know, how they behave and trying to learn something without really interfering, you know, just observational research. And exploratory research is a lot what I think many people refer to when they say data-driven research is loads of data, and maybe you do experiments, collect loads of data, and then try to learn something new about the system, right? So how would you say all of these relate to each other? At least to hypothesis-driven research first, which is, I hope, the clearest or the most important for you guys.
Hi, my name is João.
João.
Yeah.
Mm-hmm.
And I believe that the role of theory in this is like what kind of data you will collect. Because you need to have some idea about, you know, and then you're trying to collect data that can tell that you are thinking right or wrong. So maybe this.
Yeah, yes, I mean, you would use data to confirm. That's, yes, I guess that's related, I guess, to hypothesis-driven research. The way I see it, before I also complement that, anyone else wants, your name?
Beatriz.
Beatriz, okay. Wait.
Okay, it's back. I don't know, I may be wrong, but I see that, I don't know, these types of research, maybe they complement each other. So yes. I believe it's like to do a hypothesis-driven research, we would have to, like in a point, go to the data and analyze it. That's right.
And that's similar to what you said. I would say, you have Erika.
I think independent of if it's hypothesis-driven research or data-driven research, the theory plays a fundamental thing. Because if you analyze, for example, and you see correlations that might be important, how can you say that those correlations are important? You have the theory. I mean, yes, you need the theory to give like a biological background on the data that you're analyzing.
Yes, I agree. And also, you have a point? Sorry.
It's very similar to what she was saying, because the theories give you the scaffold of everything you are looking at. And you need that scaffold or that structure to know what you are doing. Otherwise, it is going to be only data without any order.
Exactly. And this is a problem that we will discuss when we talk about p-hacking. But that's for later, not now. Exactly. So I think all of this is correct. The way, for example, sorry, you have another. No, go, because at the end I'm going to tell you.
Mateus, my name.
Sorry, again? Sorry.
Mateus.
Yeah.
I think it related to exploratory research, data-driven is very important because when you want to explore some field, for example, I work with biomechanics. We have a lot of data related to movement, and we want to understand a bit about what is causing that phenomenon. So we use a lot of data, and we try to explore some theory about it, some idea.
Some theory.
Yeah. So the theory goes like they talk about. The theory goes to give meaning to these ideas. Yeah.
No, perfect. So I think we have two complementary views, at least from what I gathered. Some people say, well, of course, when you want to test your hypothesis, you collect data. And it could be very big data, right? And you have to analyze it. And that's correct, of course. In my own research, I do genomics and cancer genomics. Usually we go the other way around, and I guess a little bit with what Erica mentioned, in which sometimes we have a lot of data and we're interested in something, and we see correlations, and we try to find meaningful correlations, but then comes the hypothesis-driven research. First, you have all your data, and you come up with hypotheses, like, oh, you know, this thing is correlated. Maybe there's causality. Maybe this thing, you know, is related to, and then you have to, design an experiment to test that. Because an important thing about data-driven research, and I'm hoping that that's clear because we're gonna like say it every day probably, is that it's not final. Like you can see, this is your data set, and you can see correlations, and you can see features in your data, but you cannot conclude anything yet, because obviously there's a lot of biases that we're gonna talk about tomorrow that can be related to a data set. Right, and that's also related to your question about meta-analysis. Many people do all these studies, and is it biased or is it not? And that way we conduct a meta-analysis to try and find the truth, right? So yes, all of these, very interesting. Okay, now, how do you guys think the automation of data analysis affects the reliability of results? This is important in genomics, at least, because I work there, and a lot of things are like pipelines that are built, and things get out. Can we automate everything, or is it never going to be possible? Yes?
Yeah, I think it's related to the things that we learn in the other course. There is the when you do something automatically, you can confirm again, you can remake. Something that Andrea talked. What? Reproducibility, yeah.
Right.
When it's automatic, you can make it reproducible to the other researchers to verify your results and everything. So it enhances the reliability of your results.
That is correct. I have another view, but first, yes. Yes.
I'm Julia. I agree with Mateus at some point, but I think that we should always remember that each case is a case.
That is
and
true.
when you automatize things, like just forget that things have different meanings and different backgrounds, and put everything on the same whole, and that's not good.
Yes, exactly. I agree. That's the thing. It's complementary, right? In our own, you have a no.
It's because we have an online.
Right. And is this person okay? Like, are they hearing, and are they okay? Well, can you let me know if they are okay? And let me see. How am I doing with time for you? Okay, good. So we will have a tight schedule, so. Yeah, this is important. Before we delve into data, this is things that we need to think about. Okay. John? No? Yes? Okay, John, and then Erica?
Okay, I would just say that this process of making things automatically kind of make us think that is unbiased just by that. But someone is writing the pipeline, and this biased by this person.
Exactly.
So it isn't totally straightforward. Then it's by the computer, it's unbiased. It's nothing like that.
Exactly, and exactly that happened to, just very quickly gonna tell you that we were doing some RNA analysis, and we had a pipeline, of course, and it was automatic, and everything was nice, and there was a mistake in the pipeline. And it's very reliable in the sense that we can reproduce our errors. So, that's something that can also happen when you just there's so many steps that sometimes, sometimes, yes, this can happen. So it's important to consider. Erika?
I just
yes, I think we can.
try to connect the 2 points about Julia and Mateus and think about these specific cases. And I think that it's not like a total solution, but a partial solution. It's to be like open and document all these kind of pipelines, and in which cases you can assume that you can apply that, or in which cases it's not correct to apply that. So the very important step is to documentate.
Yes, true. And we're going to also talk about that in reproducibility. It's really important. And not a lot of people do. I mean, now it's a bigger push, of course, but still you see a lot of papers that say data on, or code on reasonable request, right? And it's not open in many cases, right? So yes, that is very important, although, for example, our pipelines in theory can be seen by people. And it's so many steps that still just, you know, it kind of slips through. So, but of course, we need to automate loads of steps because it's big data, right? So it is just now thinking, you know, how many extra checks should I do? Should I have a process of, you know, trying to go back from time to time? This is up to, you know, pretty much us, right, when we're doing these things. But, you know, just so automation of data analysis can be good because it's reproducible, can be bad because you don't think too much about case by case. You know, like Julia was saying, is that, yeah, perfect. Okay, and then another question. And this is, I think, interesting. You cannot see it, but I'll read it. What is the difference between data and noise, right? And what are data in the first place? Like, you know, are noise data? Is it important? No? No. Okay, I heard a very, like, it's not data. Who has an opinion on this? Oh, poor you have to be changing everything. What? We have more mics, okay. Good stuff. We're learning.
Yeah, so noise can be defined as the fluctuations among your observations, and it can be increased when you have less variables like replicates, and you can diminish the noise, increasing your replicates, and then you have less fluctuations among your data. So I understood as this. Of course, that can have a different...
So you would say not good, like
no. It
good.
can also reflect some important point
in your setup.
In your observation, because if you study some pattern, you can also have noise, and this system can be stochastic, so it's intrinsic of your experiment.
That's a good point. But what about if there's like actual noise where you're just, you know, maybe not looking into other, I mean, of course you can tell you other phenomena, but all biological systems will have noise, right? And if we could, I don't know, if we have a strong signal that would diminish that, would that be valuable? Because, I mean, what is the difference? I'm trying to get that, you know, what question are you interested in? I guess a bit what you're saying. If there's any other patterns, or, you know, for example, in one case, we were studying data and we had we studied mutations in DNA in our work, and there was a systematic error, and we discarded it because for us, this is not data. It's like, this is not telling us anything about our work, right? So get out. But obviously another person was like, give me all these because I want to figure out how this, you know, their question was like all our garbage was exactly what they were looking for, right? So in this case, and this goes back also to what you're saying in the sense of the data sets will have, you know, things that you can download from the internet. The people that put it there will have their own biases. And for example, in my case, I would say like, why do I upload systematic errors? Like, this is not important, right? I'm going to upload what I think is important, or I'm going to emphasize, you know, it's so much data that sometimes you make choices. And we have to be very conscious of those choices when we download the data and make sure that it's actually suited to what we want to do, right? And this goes back to this difference.
Can I add something? Yeah. Just because, I work in a very different perspective of this, but I study noise. And I study noise as a functional thing. So I just want to make emphasis. I think it depends a lot of the source of the noise. If the source of the noise is me randomly, doing like the keyboard thing, then that's not an informative noise. But if the noise comes from intrinsic to the system, then it depends on your question. Like, it doesn't matter what you're studying. Sometimes the noise can be informative, sometimes it doesn't, and you like do different levels of questions, right? So yeah, just to make emphasis of the source.
Two more.
My name is Sebastian.
Sebastian.
I want to say that, but you keep on calling it noise because you cannot reproduce exactly this point that you get out of this experiment. And it is a different view when you say data, because data thing that is reproducible even if you have a variation, but noise, you cannot reproduce again the same data point. I'm using the data word. Something like that. Leave it open.
Mariana has things to say, I can see it. Just like,
sorry. That's not always true. So the thing is, we often think about noise as just white noise, yeah? Completely random noise. But that's not true in most natural processes. If the noise comes from intrinsic to the system, it actually has a pattern, and you actually can reproduce it. And this is not related to this class, so I'm not going to get into it, but you can approach me later and I can talk about it. Sometimes, actually, like if you don't take into account the noise, the data is so unreliable that you cannot say anything about the system. But if you take into account that this is actually the intrinsic noise, you can extract information and learn something reliable about the system. So, we have, I think this is for me important because I'm not as young. Now, when we were starting college, so I'm talking about 2005, 2007, I remember very well most of the people will say that noise was this bad thing that doesn't allow us to study biology. And it was just starting this idea and a few names. They say, no, noise in molecular biology is important and is helpful. Cells live in a noisy space, so they are not fighting it all the time. They are actually using it. It's functional. And now I think everybody assumes it's true. But in that moment, there was a controversy. So I think, like this part of making that you cannot do it again, no. Noise is data as well. And you can reproduce that noisy behavior many, many times. It's amazing what you can learn from the noise. But sorry, I get very excited.
No, it's all really interesting. And noise is data. Well, I would think that noise is data also, if it's not reproducible, I would think it's still data. That happened at least once. But it's important. Philosophical questions, I don't know. We have to think about this. Okay, now two more questions and then we have an activity. And this is important, at least to me, this is very important in my work every day. And it is, how familiar must the scientists be, you, when you're analyzing data, with the protocols and instruments that were used to generate the data, with the biology that you're studying? Is that important for you to interpret the data? Or is it not? Or what do you think? This is very important to me. I study one thing and then people come and statistics, big data, data storage, all these things. Do I need to be familiar with all these things? Okay, questions and answers and opinions. Yes.
So I think that the scientists need to be familiar with the protocol and the instruments to know when the noise can be generated. For example, in my field in proteomics, if you are not able to understand how the data are generated, you can make some mistake and introduce extrinsic noise in the system. And then you have a lot of fluctuations in the data, and the way that you're going to interpret can be wrong. So I think that it's an important thing, not like a strong requirement to analyze the data, but it's an important thing.
Okay, that's one opinion. Thank you for that. What? Erika? Oh, wait. Erika and then Joan.
I was going to say pretty much what Tale said, and I'll put it a name, please correct me if I'm wrong. We are trying to model that as a batch effect, and how can we try to correct this batch effect. But yes, I mean, even if you only analyze the data, you must be familiar with how this data is generated. And that means the protocols, the instrument, and even if they come from different labs, if they come from different protocols. And yes, I will put it a name, batch effect.
Okay, nice. I'll have something to say about that. But then we first will listen to you, John.
Yeah, I would say something in the same sense, that is like, you need to have to understand the data to know when the data was good collected and good generated. And then even in some specific situations, like how to treat some NAs. You know, the NA can be above or can be upper the limits of detection, and then how to deal with it. You need to be familiar with the methods that were used to know.
These are, well, along the same lines, but now I'll tell you something. I do agree in an ideal world, right? Data in the internet that you can use for your projects many times doesn't have all this metadata. Well, that's the kind you use it. Like, for example, someone tells you, okay, here's RNA-seq from cancer samples. I kind of did more or less this. You know, I put some, whatever sequence with Illumina. That's it. It's like, dude, I don't know what you did. Like, can I use this data? Can I not use this data? I mean, this is a thing that I encounter. I wouldn't say every day in my work, but frequently, right? When you're like, well, it's RNA-seq data from cancer. Maybe I can use it, but I don't know exactly what this person did, right? So I'm more or less familiar with sequencing. But for example, different sequencing technologies, for the people that are interested in genomics, have different biases, right? As I was telling you, we had a systematic error that is just an Illumina error. It doesn't happen in other. So sometimes it says Illumina. Illumina what? Like Illumina has like 1 million different machines, right? And some of them have some. So it's very rare, probably impossible, that you actually know all of these when you, I mean, for your own data, sometimes even from your own data. I'll be honest, guys. We collect samples from hospitals to a study. I have to rely on whatever the oncologist is doing because I don't exactly know what they're doing, right? Oh, sorry. I don't know because that's their thing. They cut the tumor and then they put it to NICE. And I'm not exactly familiar with what they're doing. I know they're doing, you know, something to keep it cold. But, you know, it's an interesting question to think about until how far can you get to your conclusions if you actually don't know all the biases. I think it's, in an ideal world, you know it. But in real life, also that's why you need teams. For example, in the Sanger, I work with a physicist. And this physicist was very good at analyzing data, but he was not a biologist. And he was analyzing genomic data. And I think he could, up until a certain point, tell me, oh, you know, this gene is mutated and this gene. But he actually didn't really know what a gene was because I asked him, what is a gene? And he said, it's a box. So I was like, okay, that's an interesting point of view. But, you know, that's the thing. But he could tell me all of these lists is the most mutated genes. So, you know, it's you have to get to your questions. Have something to say? Sorry, I talk a lot, so please interrupt.
No, I was just talk about it, like the interpretation of the data, because you don't have to be an expert in biology when you are analyzing some data. It's because you can generate the graphs, but sometimes the meaning of what that data wants to say, I think this is the importance to really, so you don't have to be expert in protocols and instruments to analyze the data, but it's important you to understand what systems you are studying to create the meaning of that. So
okay, I do agree with that. Familiar is good, but you know, how familiar? Yeah.
I think this is true, and I think that that's a good thing. This incentivates the collaboration, for example. You have a biology working with a statistician or a physicist, and the biologist knows more about the phenomenon, when the statistician, the guy from statistics, know more how to analyze the data, and they complement each other. For example, the Luria Del Brook, one was more mathematician, the other was more experimentalist. So I think this is good, and you don't need to know everything. You can collaborate
on people. Exactly. So that's what I wanted to. Sorry, another thing. But yes, that's what I wanted to get at. It's impossible for one person to be an expert in everything, I think.
Yes, I think it is important to know the biology, to collect the data in nature. I think for me it's like really hard to transform the observation in nature in counting or categorical data. I think this is kind of difficult too.
Yes, exactly. So each little bit of a data analysis process has their own expertise, right? So for one person to be familiar with everything is, I think, impossible. So you do need to rely. This is why we were discussing data-driven research usually is undertaken by a big team of people, like the statisticians and the data collection people, the biologists. And then...
Sorry, just to add something. I think in general, like my experience in science is you don't need to be familiar with everything is done, but you need to be very aware of what you don't know. And I think that makes the whole difference. It's okay that you don't understand, always that you understand that you don't understand. So just to keep it in mind.
Because you can ask people, I guess. If you don't know what you don't know, then you can be like, can you help me understand? Is this okay? You know?
Well, I think that it's important to have some knowledge related with a collection of data, because when you are analyzing, you can realize when there are possible errors of measures the data.
Exactly. That relates to know. I mean, you may know sequencing technology, it has biases. And what kind, I mean, can I know which technology this was? Can you help me, another person, to interpret, you know, is this okay? So if you are aware of that, then you can go on. Like, but don't worry about, you know, knowing everything, because I think in our day and age, it's actually impossible. So you need strong teams, collaborations, and of course, know what you do, right? Your stuff. But if you are, for example, a PhD student and you have to oversee your whole project, being aware, you know, of protocols exist, different protocols exist, they have different biases. Which one is this one? Oh, it can have these biases. I may consult an expert on these biases and, you know, can I continue? But I would say, yes, of course, you need to be familiar, but you cannot be an expert, you know, and knowing everything, I would say. And this is also interesting. We use lots of programs this day to do things, right? You know, everything. People use papers all the time in protocols and stuff, you know. How, to what extent really can these algorithms identify causal links in data, right? Because they will come up with mutated genes and your disease state, but, you know, can you rely on that? Is that, you know, there's many algorithms as well. Do they agree? Like, what would you say here? And from here, we'll go to an activity after that. But I'd like to hear opinions on this. It takes a while, right?
Yes. Well, I think that those correlations, whatever algorithm you're using, statistical correlation we are applying, without any like subjacent underlying knowledge about the system, you cannot make causal relationships between things. And even with a good knowledge of how things relate phenomenologically, even so you can like give some educated guesses and some statements that you can like put some pressure and say, okay, I think this is fine. True. But even so, I think you cannot like deduce that's one.
Sure. Yeah, no, exactly, because you cannot rely on algorithms to find things. You still need your knowledge, right? And then on another day, I can tell you a story how I learned this the bad way. But it's true. We cannot just rely on pure, statistics or algorithms, we also need some knowledge, right? To know, of course, that comes with biases as well. So it's a balance between our statistical approaches and our knowledge of what many people have discovered. Science is a thing. Like, people have been building it for years, and we cannot just ignore it with our data set and our statistics, right? Okay, now we have an activity, and of course I've talked for a very long time. We are running late on the first day, yeah, I think we can have 10 minutes for this. I think that's fine. Basically, there's teams, and you cannot see the other team because there's a screen there. But we've made teams. What Mariana did these teams based on your expertises, trying to make it, varied. So would be okay, guys, if you get together in these teams and just write down here, what is a gene? Don't look it up. Just come up with what you know. What is a gene? Define a gene and put it here. Oh, no, wait. How do I do this? Well, go there. It's a Google form for you to just... sorry?
We can discuss and rewrite.
Yes, exactly. Discuss, and then just write what you think is a gene. Come up with a... Yeah, of course. Go.
1 minute, guys. Start typing.
Start typing. We have
1 minute. I think one team is missing. Oh, no, there it is. Everybody. Perfect. Okay, so I'm just going to read it you really quick, everybody's answer. So first, codifying functional part of the DNA. Codifying functional part of the DNA. A transcribable section of DNA that can be inherited. Sorry, something happened here. Any DNA sequence that can be transcribed, a sequence of nucleic acids that codified a metabolite, genetic information unit of gene product fabrication, sequence of nucleotides encoding the instruction of a specific trait.
Really cool. Okay, thanks, Pollo. So what did you see in all these definitions? Some people said certain things. Sequence, okay. Transcribable, information inherited. Okay. Right, so, but why, I mean, if gene is a word that we use all the time in genetics and all this area, why was your definition not the same? Like some people said metabolite, some people said transcribable to something. Does it have to be transcribed? I don't know. There's certain sections of genes that maybe are not transcribed. I don't know. But this is actually the exact point of this activity. And basically, it is that biology is notoriously fragmented in methods, in goals, in instruments, conceptual frameworks, and definitions. Right? This is exactly the point. Like your definitions have something similar, but for example, some people said inherited. People didn't say inherited, right? So what really defines a gene? Or anything that we discuss in biology. Now, this is, and we're going to share this paper for you. This is a really interesting paper, I think, where the authors, which, you know, we're here, but you can see. Well, anyway, we're going to share this paper. Basically, all the different definitions of gene that still exist to this day, right? And it has evolved, of course, since the 1920s, when the word was first used, when first it was a dimensionless thing that was, you know, the heredity something, because it wasn't even a molecule, it was, you know, the basis of heredity. And then it acquired, you know, like a one dimension, it was long, it had length, you know, and then some people said, well, it's continuous, and then some other people said, well, actually, it's not continuous because it has introns, and then people said, but the intron is part of the gene, and then they said, but it's not transcribed. You know, does it have to code for a protein? Some people say, well, you don't have to code for a protein because there's long non-coding RNAs. Are they genes? Are they not genes? You know, all this, this is an interesting question in general. We just wanted to illustrate it with, you know, this word, gene. That is pretty much the most used word in genetics, probably. But this is a feature of biology, and it's especially important in data-driven research because people will write words in data sets. And they will say things like metabolism, and it's like, what does metabolism mean for this person? And they wrote their experiment and they described it like that, and do they mean the same thing that I mean? And this goes back to, can I use this data set? Did they mean the same thing, right? So I mean, of course, I'm not expert in any other areas of science, but I would think this is similar in many areas of science that rely on a lot of data, because definitions are fragmented and people mean certain things. All your definitions were correct, but they were not the same, right? And if you put it in, describing a data set, another person maybe this is not a gene. Maybe this is, something different. I cannot use this data when you maybe meant the same. It's a big discussion. But this paper is interesting, and it goes over all the definitions. These are recent and how it has evolved. Sometimes they add a functional dimension to it, like it has to do something, a gene needs to do something. Some people say, well, no, it just needs to be inherited or transcribed, but it doesn't really have a functional meaning, right? So this paper is interesting, goes through the history. We have it in some Dropbox thing that we're going to share with you. But basically, well, that's a challenge. Now, data generation, which we will go over tomorrow, also has its challenges, because instruments, of course, if we were designed, are built to satisfy specific objectives. As I was saying, for example, in my case of my systematic error, I don't want it, it's not satisfying my research agenda, and I'm uploading the data anyway. So we need to be conscious that observations and measurements are always situated in a specific framework. That means that no data is really raw, because people have already tuned these instruments to put something out specific, right? So you have to be conscious of that when you also download things or are not exactly familiar with how your data is generated. You may be able to use it, but you have to be aware. As Mariana was saying, be aware of what you don't know. It's important. Okay. Now, another very important thing here is data quality. So this is, again, when you use big data, usually you obtain it. I mean, you're not going to generate all the data, probably, that you will use if in a big data project. Maybe you will, and we will see some examples of people that do generate in their own labs, like vast amounts of information, like single-site data and that kind of thing, right? That's a lot of data. But usually, if you want to use your collaborators' data or something that you downloaded from the internet, data quality also varies vastly, and it's not raw. Again, people are like, these are reads from the sequencer. Like, yeah, well, how is your sequencer tuned, right? Or, did you put all reads? Did you just did a little bit of curation? And sometimes they don't tell you that. That's the thing, right? It's very hard to find this in the metadata of these projects, right? And this is interesting because these are often specifics of certain fields of research, even within biology, right? So, for example, some people will, even in my area, that is genomics, sometimes I don't want to do quality filtering because I'm interested in everything, right? And then I'm going to select something and then I'm going to test it, right? So maybe I can upload a data set that is really not curated at all, right? And even if, I may also have questions like, now I want to find a specific genome mutation. In that case, I need to do data filtering, right? And what the person that uploaded the data was thinking is something that you also need to be conscious about when you use data from other people, specifically. And then something that we're going to talk about in, I think it's class 7, reproducibility. It's important to ensure that our results are robust and reproducible across datasets. Again, meta-analysis, that was mentioned, that's important for this. But it is one of the biggest challenges of modern biology. And we will see also an activity where some people have claimed that most research findings are false. So, we will discuss that, and we'll see if you see some merit in those claims. Many people have many opinions on this. Okay, so I think we've finished for today. This is just a summary of what we've talked about today. So first, what the definition was: data-driven research seeks to extract scientifically interesting signals in your data. For that, of course, you do analyze very large data sets. That's what the definition of data-driven research is. But you need statistics and many other tools in order to fulfill your goal. And sometimes this is what it means that you need a big team. You cannot be an expert on everything. Also, that data-driven and hypothesis-driven research are different approaches, but they're complementary. Of course, at least how we use it in our data, we use the data to generate hypotheses, and then we design an experiment to test them. But in other cases, of course, you can have a hypothesis-driven approach and generate loads of data and then do everything that we talked about, right? Cleaning and doing all of those. And finally, well, what we were discussing now, we need to be very careful with definitions, basically, of what people mean when they upload things to, data sets. Metadata, what exactly does it mean? These words, gene metabolism, are used in many, many different settings, and knowing exactly that you're getting what you are getting is a big challenge for your data and for biology as a whole. And then, of course, be mindful of data generation techniques. They vary. They are subject to whoever generated the data, what their agenda was, and the data quality processes that we at least know what we're downloading. Sometimes we assume it's filtered and it's really not. Any questions about?
No. Sorry.
Before
No
opening
questions,
to
questions, just in the matter of time. Can you so the person online can see this? So just to explain really quick. We are, as part of the activities, every class, we have some questions at the start of the class that are going to be concepts that we are going to see in the class. So it's not the idea that you already know the answers. And then we repeat the questions at the end. We forgot today at the start of the class, so it doesn't matter. But here's the link for the end. Please answer it. It is more about participation, more about to see what you think about what we, like the concepts that we try to explain, that Daniela tried to explain during this class. So don't worry too much. It's really quick. It should only take like a couple of minutes. Don't think too much about it, but please do it. And after that, we can open to questions. And meanwhile, I just wanted to mention the teams that you work today are going to be the same teams for all the activities, the rest of the class, except the final presentation. So if tomorrow you want to sit, like clustered, it can be useful in terms of times. But answer these really quick, and then we can open to questions in the last few minutes.
Think of your questions. I will do that then. We can answer at the end. Thanks. and also if you have any questions about today, now is your time. Of course, we're going to be here for 2 weeks, but if you wanted to say something about today, or was it all clear? Was good? Guys, all good? Yeah? Okay.
So if you submit your questions and you want to go, you can go. If you have questions, you can just tell us now, approach, whatever you want. But if not, that's the end of the session today.
And tomorrow we'll see data generation.